I'd like to welcome everybody. I'm Marcia Stefanik. I'm professor of medicine at the Stanford Prevention Research Center at Stanford University, and also professor of obstetrics and gynecology, and the director of the Wisdom Center, which is the Women's Health and Sex Differences in Medicine Center at Stanford. Today we have an amazing speaker presenting on sex and gender matter, now what? Incorporating sex and gender in biomedical research, and that is Sabine Oertelt-Prigione, who is the strategic chair of gender in primary and transmural care at Radboud, Nijmegen, the Netherlands, and also professor of sex and gender sensitive medicine at Bielefeld University in Germany. I'm going to stop sharing and so that we can actually see our speaker and see me. I'd like to say a little bit more about Dr. Oertelt-Prigione. She is a physician, researcher, organizational consultant, and coach. She's the chair of gender, I already said that. She's a member of the EU Commission Expert Group, Gendered Innovations 2, which is actually chaired by Stanford's professor of history and history of science, Londa Schiebinger, and she's currently chair of the expert group Gender and COVID-19. Her current research focuses on the implementation of sex and gender sensitive research through the analysis of practices, development of methods, and engagement of stakeholders. Her current research focuses on the implementation of sex and gender sensitive research through the, oh, sorry, I just said that. She has developed several open access resources for sex and gender sensitive research, spanning from databases to open access books and methodological toolboxes. In addition to her academic activities, she advises healthcare and research organizations in the implementation of prevention measures against gender discrimination and harassment. I've read many of her works and she's an outstanding researcher. I can't wait till we get to hear her. So with that, Dr. Oertelt-Prigione will share her screen.
Well, thank you very much for this very kind introduction and for giving me the opportunity to be here today. So I'm very much looking forward to, well, telling you a few things about what I do, and then of course about an engaging discussion. I also have some personal ties to California, so it's nice to be there virtually. I lived for 3 years there, actually got married in San Francisco, so there are some personal ties. So it's really nice to be able to be here virtually, and I hope the next time I might be able to join in person.
And I want to just interrupt for 1 second and say that we believe that you can ask questions in the Q&A and that we can answer them. So there should be on the bottom of your screen a Q&A function, and feel free to put questions in the Q&A.
About that, unfortunately, I will not be able to see them, so feel free to interrupt if there are any direct questions. So if anything should not be clear, otherwise we can address them in the discussions, because unfortunately I will not be able to see the Q&A section or the chat at this time. Okay, so what will I be talking about today? Yes, about the fact that sex and gender matter, and most importantly about where we are currently in the incorporation of sex and gender as biological variables, and how far that affects the work that we do. So I will start with a brief question about why should we consider sex and gender at all in biomedical research, and I'm not going to answer that personally, but let our funding organizations in the United States and Canada and Europe answer that question. And then I will move on to the larger part about operationalizing sex in biomedicine, how far we can do that and how far it is being done and what we should consider. And then move on to how we can operationalize gender in biomedicine. I'll give a few examples of how this can be done in practice. And then I will conclude with a brief section about how content and context are actually connected in this field. So the main question, I think, that moves a lot of us and a lot of the researchers that are doing it or not doing it is, why should we consider these aspects at all? So why does it matter to look at sex and gender in biomedical research? And by using the words of the research funding organizations, well, it's not some small things that we should be addressing here. It's really big topics that we are supposedly able to do much better if we consider sex and gender. And so in the words of the European Commission, what they expect is that by looking at sex and gender in the work that we do, we will increase and improve the scientific quality and the societal relevance of the work that we perform. The Canadian Institutes of Health, especially the Institute of Gender and Health here, say that it increases the rigor, the reproducibility, and the generalizability of the work that we do. It's about excellence, and the NIH says that it's about, again, increasing the reproducibility through rigor and transparency. So, as I said, it's not some small parts, it really goes to the core of a lot of the aspects that we've been discussing in the last 5, 10, 15 years, also in the field of science overall, especially in consideration of some reproducibility issues. So we really see an opportunity to increase the quality of the work we do, do excellent work, and of course, also increase the societal value of the questions that we are addressing and the answers we're trying to provide. I will use the model of the Horizon Europe of the European Commission here because it nicely ties together two layers that we actually have to address or can address in this field. And when you look at how the European Commission operationalizes gender, or the gender dimension as they like to call it, there are really three aspects to this. So the first one is the gender balance in the research teams. The second one is gender balance in the decision-making processes within science, within these research teams. And the last one is really the integration at the content level of sex and gender in research and innovation. So if we have to sum this up, there are really two layers to this. One is the context layer and one is the content layer. So given the time we have today, I will only focus on the content part. So I will talk about how we do this and how it affects the work we do. But I think it's important to acknowledge that this context aspect of who is doing research, who's asking the questions, and which kind of balances are available in the field of science impacts the questions that are being asked and the topics that are being given priority. So let's move on to our content part. I will start with this as a sort of small summary of the terminology we like to use and we are mostly using at the moment, which brings together these two layers that I will be addressing today. What we see here at the core is sex, and with that I mean the biological differences, physiology, anatomy, all these aspects, and I will go into detail of how that matters for research. And what you see around this are really different dimensions of gender. The main and probably largest question that we have to address is how do we operationalize with the methodology of biomedicine, which is mostly quantitative, how do we operationalize a concept that has been profoundly developed in the social sciences within a context that is quantitative in nature? How can we develop instruments that allow us with our methodology to actually capture a concept that is multidimensional, that has several aspects, and that can be defined also as fluid and not binary in nature. And I will try to give some examples of how we are doing that at the moment. Now, we start with this, with the fact that every cell has a sex. So looking at sex is not just relevant for sexuality and secondary sexual characteristics, but it is really affecting all the cells we could potentially be studying at the cellular level when we use animal models or when we're looking at humans. There are really three layers that we mostly use in operationalizing sex, which are the 3 Gs. The genetics, gonads, and this is a proxy for hormones here, and genitalia. So these are the 3 layers that we are given to operationalize sex. When it comes to genetics, this is probably the one that is mostly used as a dichotomous concept. We have 2 sex chromosomes, the X and the Y. XX is generally attributed to female, XY is generally attributed to male. The assumption is that the population divides into a 50-50, which is true up to it's almost fully true, but we know there are individuals that either have a X0 karyotype or multiple copies of this X, so it is not a full dichotomy we could be working here with. But what is even more important and maybe more interesting is the fact that throughout our lives, and especially with the aging process, it is well possible that in peripheral cells, so in circulating blood cells, for example, we find monosomy, generally of the X chromosome. And so with the aging process, as the older we get, it is more common to find cells that have a monosomic karyotype circulating in perfectly healthy individuals with a general XX karyotype, but in these peripheral cells, at least in these cells that's been described in fibroblasts, in blood cells, we can find it. And it can be a marker for potential immunological vulnerability. It has been associated with autoimmune diseases, but it is not a given marker. So not everybody who has cells with a monosomic karyotype among their blood cells will automatically develop an autoimmune disease. But that's just to say that there is a certain dynamic to this, and having an XX karyotype or an XY is not fixed in all cells at all times throughout our lives. The second layer we could be looking into are the hormones, the gonads. Interestingly enough, we mostly work with cutoffs here. So if we consider that almost everyone, apart from people who have selective inability to produce certain hormones or have resistance to certain hormones, almost all of us produce all of these hormones, but they are of course distributed in different way across the population. What is interesting and important here is that hormones, all of them, vary throughout the day, throughout the month, throughout our lives. So, and that doesn't just go for the menstrual cycle, for example, or for menopause. It also goes for testosterone. Testosterone levels are different in the morning and at night, and they vary every day. So this is important to keep in mind, and the study of hormones is not just essential, especially when we look at reproductive questions. It is also essential when we look at a lot of other cells, and as I said initially, every cell has a sex. And here, for example, I have an example from the immunological field, where we see that depending on the phase of the menstrual cycle, and hence which hormones have higher concentrations, we will find a different response of the immune cells. So in the follicular phase with high levels of estrogen, it is more likely to have a Th2 response, so more production of antibodies. In the second phase, in the luteal phase, when progesterone is higher, we're more likely to have a Th1 response. So for example, in experiments in the immunological field where we try to predict T-cell responses, this is an essential aspect to keep in mind as a modulator of Th response. The third level, and that's pretty much how most of us have been assigned to one sex or the other, is actually through anatomical observation at birth. The baby is born, we observe genitals, and the child is assigned usually to either female or male. This works for large groups of individuals, although there are certain people where, for example, the external and the internal genitals don't match, or where a genital cannot be clearly attributed to one or the other. This can be due to several reasons, and depending on the studies we're looking into, the prevalences are quite different. So it has to do with the fact of who is defined as intersex or defines themselves, identifies as intersex. And that means that we can go from percentages from 0.07 to up to almost 2%. So just to say that this is not an absolute rarity that happens one in a million. Why did I put a mole here? Of course, moles are nice and cute animals, but moles have, and I didn't know that until a few years ago, this was actually researched by some colleagues in Germany who found that female moles are always intersex. So the female moles, while having a normal functioning ovary and being able to reproduce, also have testicular tissue in their womb. And this testicular tissue is responsible for the production of testosterone, which they need to develop appropriate muscle mass to be able to move properly under the soil. So they are naturally intersex, just an example of a mammal that has two biological correlates normally throughout their lives, and it doesn't impact their fertility. Why is it important? Now, we have three abilities to define sex, and they are all a bit more complex than they might seem at first, but why does it matter, and in how far can it affect the work with cell cultures, for example? Well, first of all, in many cases, we don't know the sex of the cells we're working with. And I like to ask this question when I do these trainings in person and ask people, well, do you know the sex of the cells you're working with? Some people do, a lot usually don't. And that's one of the first questions that might need to be addressed. And it's also true that, for example, some commercially available lines, for example, fibroblasts, are only available as male cells. They're not available as female cells. Then, as I said before, karyotypes are not stable, especially when cells are kept in culture for a long time. And we know that from the field of tumor research. So these cancer cells that have been kept in long cultures, like think, for example, about HeLa cells, we know that a lot of times the karyotype changes over time. And that doesn't just go for sex chromosomes, that goes for all chromosomes. But that also means that if you're working with cell cultures, you might have some anomalies in these cells that you might not even be aware of. We know that certain techniques, like, for example, reprogramming, change the way that the X chromosome is made up. So, of course, you need to de- and remethylate the X chromosome through the process of reprogramming, which will change its epigenetics. The type of media we're using can have an impact. And we don't know if, for example, methylation, since there is an intergenerational effect of epigenetics, so if the profile of origin can actually have an effect on that. What is still open questions that need to be addressed more in depth is in, for example, the hormones we use for experiments, if there is a difference in the synthetic or potentially purified hormones, so if these work differently, and then if there are intrinsic differences depending on the reaction to media. So do cells that are XX potentially show different growth patterns than cells that are XY with an average growth medium? So there are a lot of questions that go in the direction of basic methodology that are still to be answered. This is a very nice review that I would recommend highly if you want to look at how you can approach, for example, the study of sex differences in animals. Frank Movey-Jarvis laid it out for you, and basically you have to start at the first level in looking at female and male animals, looking at whether there are differences, and if you do find differences, you want to find out where they're coming from. So the first step that you're going to do is look at whether it depends, for example, on ovarian estradiol. And what you're going to do here is you will do a series of experiments to see in how far the estrogens are actually responsible. So the female mouse will be undergoing sham surgery, first of all, as a control, of course. And then you will go and proceed with ovarectomy. And on the one hand side, replace these estrogens to see if the effect that you're seeing in the natural wild type mouse or in the bred mouse that you're using here is reproduced. And the second one is doing it with a vehicle that, of course, is not estrogen to see if the effect disappears. And then if you find these differences, you can confirm them, you will have to go on and find out which of the estrogen receptors is actually responsible for that. So is it the estrogen receptor alpha, the estrogen receptor beta, or the G protein coupled one? And once you identify that, there are several knockout mice to do that. You have to go on, ideally, and find out which tissues are affected and in which form. So as I said in the review, it's laid out in detail how you can go about this, but it also shows that there is an entire procedure to actually identify what the effects are specifically in the specific organs. That's just one example. And I think it's also important to point out that although we think that it is possible to control everything we do with the experimental animals we're working with, some of these things that we control, some of these factors that we control can have potentially sex-specific effects. So for example, the diet, if you're giving soy-based chow to the animals, soy is an estrogen mimetic. So that might have a different effect depending on what you're giving them. The social dynamics in the cage also play a role. We know that mice are social animals, rats are social animals, and they do develop a certain social order, which is different for the females or the males. There are still questions about how far temperature plays a role, for example. We know that in humans, we do see some differences in reaction to temperature and what feels comfortable. Lightning and circadian rhythms might also play a role, and I will come back to that later. Research staff might also potentially play a role here. And then, and this is the last one for the animals here, we should not forget that there might be intrinsic differences that we're simply not aware of. And this is work from colleagues from Barcelona and Spain who have published this last year. And when I talked to them, they said, well, we were so surprised that nobody ever looked at this before. So what they investigate is memory formation, and they look at pain models to see in how far pain leads to memory formation and what kind of factors influence memory formation. And what they were studying was the neurokinin pathway, which is relatively well established in its effect on memory building. And they were looking at antagonists of the neurokinin pathway. And what they saw was, which was known, if you antagonize this pathway in the presence of testosterone, you can actually interrupt the memory formation process. However, if you use the same antagonist in the presence of estradiol, what you actually do is you potentiate the memory building process. So this was quite surprising to them, especially because they said this has been investigated, these processes have been investigated for a while, but this really different reaction to an antagonist, depending on which hormones are present in the environment, was never documented before. And of course, it has an essential role in how we can study memory. So that's just to say that even for processes where we think these are physiological, we have disentangled how they work, if we have never looked at potential sex differences before, there might well be some that we need to investigate and make clear. Well, if that showed that investigating sex is not as simple, as straightforward as it might seem, let's move on to gender to make it a little bit more complicated. As I said initially, we are of course trying to implement and to investigate a variable that is not originally defined in the biomedical sciences. So what we try to do is we try to divide it in different dimensions and try to investigate these dimensions separately in order to get a good concept of the different effects at different levels. Of course, what we do here, and this can be discussed and is discussed, I would say, with colleagues in the social sciences, is we are kind of fixing the concept. So we are moving from a dynamic nature of the gendering process, which is doing, to actually a forced picture in time, which is being, as if gender were not situational. So this is, of course, something that is difficult to capture when you use a questionnaire. So let's start with the first layer. The first layer is probably the one that's mostly discussed in the media, which is helpful and sometimes it's not, because of course it brings attention to the fact that we need to consider gender and its impact on healthcare, but it also a lot of times limits the discussion to the identity aspects and really doesn't go to the core of all the other factors that play a role. When it comes to identity, what we tend to do right now in several of the statutory cohort studies all over the world are starting to pick this up. Several, also the ENHANCE, for example, in the United States is picking this up as well, is to divide the question about sex and gender into two. So use a two-step approach, which allows to investigate sex assigned at birth, which is biological sex that I've been talking about for the last 20 minutes. And then the second question will address gender identity, current gender identity, knowing that gender identity can change throughout life. And of course, that it is something that we need to ask people in order to know what it is. I cannot define it from outside. Why is it important? Well, first of all, because in medicine, we tended in the past, and sometimes people still do, to conflate the two. So a lot of times when we look at biomedical studies, we don't really know what had been investigated. We assume that in most cases it's biological sex. Maybe it was gender identity. So in many cases, these two terms are conflated. So that's the first layer. But then for a lot of research questions, we might need both of these pieces of information. So it is really important to have both of them. For example, if you investigate pharmacological side effects, you want to know about sex differences because they will possibly be relevant, for example, to the metabolism of the pharmaceuticals you're taking. And on the other hand, you want to know about gender because who is reporting these side effects? Are they being taken seriously? Are these being recorded? Are we acting on that? So that's more connected to gender identity and maybe gender relations. So even in a study that looks at a process that is generally seen as strictly biological, you might need both these pieces of information to be able to actually properly investigate it. The second layer that we can look into is gender roles, and I show here the BEM sex role inventory, well knowing that it has been discussed and it has been questioned for many years, since it was developed in the 70s, actually around the corner in Berkeley. But it is still the most widely used instrument to measure gender roles in biomedical research. So regardless of all the criticism about its ability to represent some of the dimension that it set out to represent, it has been used more than 120 times worldwide to investigate gender roles in the biomedical field. We have several other instruments. I would say at the moment there's none that is absolutely perfect. So all of these have their advantages and disadvantages. What is maybe important to point out here is that the essential merit that I would attribute to the sex role inventory was moving the discussion about masculinity and femininity from a one layer dimension to two dimensions. So before the establishment of this discourse by Sandra Bem, the perception was that masculinity and femininity were two poles on one axis. So you were high on one side, you could not score high on the other side. What she did with this instrument was to actually disentangle the two. So there is one dimension for masculinity and one for femininity, which allows every one of us to potentially score high on both elements of femininity or low on both elements of femininity and masculinity. How these elements are defined, we can discuss later if we want, but it really allowed to show that, first of all, gender identity was disconnected in a way or another from gender roles, and that there are multiple layers that affect each and every one of us, and there's not just one polarity. So that was definitely the discussion that was initiated through this. When it comes to gender relations is really, I would say in simple terms, how identity and roles, so projections from outside, interact. Gender relations can be measured at several layers. You can measure them at the private level, you can measure them at the professional level, and you can measure them at societal level. Most of the work that has been done in connection to topics of health is at the personal and private level, so in personal relations, although there is some at the professional level. So examples of this is at the personal level. Well, for example, if children are sick, who's staying home and who's going to work? Or at the professional level, if you have to negotiate your salary, in how far does gender impact those negotiations? This is one example from this private domain. It's a study from Sweden. And here, heterosexual couples were asked about the gender equality in their relationships, how they would rate that. And I show this to you because it is really interesting in showing us that when you are investigating these aspects, you should always consider at least two different methods. Because the first one was the self-perceived gender equality. So this is the brown bar. And you see the brown bar differs quite a bit from this peach-colored one, which was an index which was constructed based on, for example, hours dedicated to domestic work, unpaid care, who takes the children to the doctor when they are sick, who stays home, and so forth. So it was an index built on many other objective measures where hours could be defined or specific actions could be defined, and then these two were compared. And this is just to show that there is a very strong social desirability bias, especially depending on where you're living, and this was done in a Nordic country. And so if you only ask people about their self-perceived gender equality in their relationship, you might get quite different results than if you measure it. So that's just to say that this is really one where you need triangulation. How are we proceeding in the field? So since I would say, well, the mid-2010s, around 2014, 2015, our colleague Luis Pilote in Canada has come up with the gender index, which was the first of a series of different indices that are being developed with the objective to develop instruments for biomedical research. All of them in one way or another try to bring together different dimensions of gender. So I just showed you these dimensions separately, For example, in this one, as you see again, the BAM sex were inventory, but then when you look at these, for example, the hours spent doing housework or the status of the primary person responsible, these are gender relation factors. So it is a combination of different questions to develop an index. What you get in this case with this gender index is the probability between 0 and 100% to be a woman. This has been discussed if this is, well, the best way, in quotation marks, to operationalize it, especially if we think about what Sandra Bem did with going from a unidimensional construct to having several dimensions. But it is definitely, and we definitely have to credit this index and the following ones for reactivating a methodological discussion that we definitely need in the biomedical field to make much more clear what it is that we're researching and also allow for more robust results in the future, and possibly also for more engagement with colleagues that might not be so convinced that this is helpful. So we definitely need instruments that are applicable. Now, it is not easy, especially when you are dealing with human beings. It's a little bit easier when you are working with cells. As I said, with animals, there are already some tricky aspects in their social behavior, but humans live in a world outside where you cannot really control a lot of the things that they do. And so what that means is that really sex and gender interact throughout our life. And we start here, and these are the 3 Gs, the genes, genitalia, and gonads at birth. And then we grow up in a gendered system where at the family level, the community level, the institutions we go to school in, study, and work in, at the structure and policy level, there are always gender norms and gender roles. We live in a somewhat gendered world. And all of these aspects influence our positions in society. And what you see here is also a concept of intersectionality where gender is already portrayed in interaction with other social domains. And that, of course, leads to different pathways in health, to different interaction opportunities and possibilities and consequences in the healthcare system and in the healthcare we're providing at the individual level and at the population level. So I would really invite you, if you're interested, to read this very good review. It's been published 2 years ago, which exemplifies nicely how the two concepts interact with each other. Now, after this really long, I would say theoretical part on how we operationalize sex and gender, I would move on to some of the consequences. So what can go wrong if we don't do it? And I'll also talk a little bit about a few of the things that we've been doing lately. Well, the first part, and I briefly mentioned that, but I didn't go into detail before, was, we always think we can control everything when we work with animals, where most of the things these animals are exposed to are really under our control. But it turns out, as this paper nicely exemplified several years ago, that also the person performing the research can play a role and impact the reaction of the experimental animal you're working with. In this case, it was, again, a study in the field of pain. And what the researchers found that really the pheromones of the researchers handling the animals, the mice and the rats, had an impact on their reaction. So the way and the amount of pain they showed differed depending on who was the experimenter handling these animals. And of course, this opened up a whole new discussion about potential trans-species perception of pheromones, because in general, we assume that pheromones are only relevant for members of the same species. But what we see here is that really the human pheromone is also picked up by the experimental animal. And that's definitely a question of reproducibility. So how do we need to report that? Do you need to report it? And in how far could we control for it? The second risk, and this is a panel of proteins that has been investigated in the field of depression and anxiety. So it's a fixed panel of several proteins that have been correlated with the risk of developing anxiety and depression. It's a study of about 2,000 individuals that have been done in the Netherlands. And what they showed quite nicely is that depending on the moment of the menstrual cycle the participants were in, or on whether they were taking oral contraceptives or not, or whether they were postmenopausal or had high testosterone levels, you see that these proteins allocate quite differently on the panel. And interestingly, what they found is that if you don't match your controls properly based on the phase of the menstrual cycle, or for example, the assumption of oral contraceptions, which we don't always do in our study, several times we don't even know if people are taking hormones in any form, not just oral contraceptives, it can give very skewed results because they do affect the protein production in this case. This is probably one of the most well-known risks that we incur if we don't consider this. We know that several diseases are attributed to either one sex or the other, I would say, actually attributed on gender identity, probably more than that, or they are associated with certain symptoms that we learn in medical school. Now, it turns out that this can be problematic, and several examples are, for example, bladder cancer is notoriously, the delayed diagnosis is quite common in women because if they report hematuria, we first think about a urinary tract infection, which makes epidemiological sense. But if it is not a urinary tract infection, we tend to be a bit too late sometimes in starting investigation for other potential diseases. When it comes to asthma, for example, we think about typical wheezing. The wheezing is much more common in young boys. In young girls, a lot of times they only report dry cough at night, which is not immediately identified as asthma. When it comes to diseases, there are typically women's diseases. So for example, osteoporosis, which we generally attribute to postmenopausal women. Men are not screened systematically, although 30 to 40% of over 70-year-olds do have osteoporosis. And then this is a little study from Israel, which is quite defining in a lot of care pathways. This was about multiple sclerosis. So multiple sclerosis is an autoimmune disease which is notoriously difficult to diagnose because a lot of times you might have a first flare and then for a really long time, sometimes a decade, patients don't have any symptoms. So the disease is not necessarily suspected. And here we asked, or not we, the researchers asked the patients, well, which kind of specialists did you meet in your journey before you were diagnosed? And a lot of the men said, well, at a certain point I was sent to an orthopedic surgeon, while a lot of the women said I was sent to a psychiatrist, which usually didn't happen to the men. This, again, is also one of the examples that we know quite well, and yet we are still not doing it properly. This is about side effects and pharmacological efficacy. It's a historical study again. This is from the 90s, the DIG study about the safety of digitalis. What you see here is digoxin is first tested against placebo, and when you look at this, looks fairly good. Doesn't seem to increase the risk any more than placebo. So digoxin was then widely employed, mostly for heart failure. And 5 years later, after a large number of women developed side effects, these data were analyzed again. And it turned out that actually in this group, digoxin at this concentration was quite effective in men. It was more effective than placebo, but in the women at this concentration, the placebo was actually safer than the digoxin. Why did we not see that? Well, because women were 25% of the participants and men were 75%. So once you put them all together, the answer you got was this. And this should be one of the clearest examples of why we need to disaggregate data. And yet, this is data from last year. Together with some colleagues, we looked into how it was going with the COVID-19 trials, assuming that we had learned from the past. And unfortunately, when looking at the registration for clinical trials between January 2020 and January 2021 for COVID-19, we looked at more than 4,000 trials, and only about 20% explicitly said that they would consider sex and or gender as a recruitment criterion. Only 4% actually mentioned it in their analysis plan. Of course, the registration is early on and everything could go much better. Maybe everything changes. Things do get a bit better, but once we looked at the published trials a year later, about 20% actually contained disaggregated analysis, and several colleagues independently from us have done other studies in this field, and they all find around 20%. So they did reproduce the same numbers we were finding in the publications. That's just to say that things are getting better, but up to a certain degree. Now, the last 2 points. how can we deal with this in current studies or with data that we already have? I've been talking mostly about sex differences here, especially because in the field of biomedicine, we tend to not have a lot of information yet about these gendered aspects. And we try to look at data from the oncology field with a slightly different angle. So this was a collaboration with colleagues that have a very large repository of data. They observe patients that are cured clinically from cancer, that are long-term survivors, and they have a very large database in the Netherlands, and they follow them up for 5, 10, 15 years. So all of these participants had been cancer-free for at least 5 years, and we looked at how they were doing. And what we saw was that the female patients had quite predictably this matched data that we had already seen in the literature in smaller cohorts, more symptoms persistent even 5 or 10 years after being cancer-free. You see here nausea, pain, insomnia, although depression, for example, was more common in men. However, the interesting thing we looked at was in the health reported quality of life questionnaires, you also have several dimensions that are being asked. And what you see here, they ask about functioning in the physical domain, the social domain, the cognitive domain, the emotional, and the role domain. So these are aggregated questions of multiple items to define general functioning. And what we found here is quite different than what had been found in the literature before. First of all, because it has never been investigated in this form, and because usually no comparison is done with the general population. So when you look at studies on quality of life, most of the time the female patients are compared with the male patients. In this case, we had a control group from the general population, and since the quality of life, especially of the female for the female control group also decreases over time, once we compared the impact of cancer, of long-term survivorship in the female and male patients with the general population, it turned out that the role functioning and the social functioning in the male patients decreased significantly, actually much more than in the female patients. So this is quite different from before. And if you think about the different gender dimensions, this role functioning can be seen to a certain degree as a proxy for gender roles. And this is not explained by loss of employment. So that was one of the first things we thought about. It is not justified by loss of employment. It is actually, it doesn't impact at all. So that's just to say that even with data that's available, sometimes if you look at it from a different angle, you might find gendered factors or gendered aspects in there that haven't been investigated. But it is important to have control populations. Again, this is work in progress. It's under review right now. Here we tried to investigate the different gender dimensions separately. So in this study with patients with Parkinson's disease, with people with Parkinson's disease, we are looking at different ways to investigate gender. And we have a quantitative part, which is what I'm showing you here. There's a qualitative part, and there's a design-based part where we try to investigate in how far design-based methods will allow us to access different questions, like, for example, the emotional aspects connected to this. But this is just to show that if we use a gender index, we risk losing some of the details. Because what we found here, we looked at identity and roles and relations and several other factors. When we looked at identity, when it comes to the health-related quality of life, we didn't find any difference. And yet when we looked at roles, we saw that the ones that scored high on androgeneity, so high on femininity and masculinity, actually did much better compared to everybody else. And then again, when we looked at relationships, and this is quite interesting, but I cannot show it yet because it's not published yet, in the relationship domain, it actually turned out that the ones that had equal division of labor at home especially had a higher quality of life, which was quite interesting to see. It has several possible explanations behind it. But what it shows is just that these different dimensions can give us different information, which has a different impact on the clinical recommendations that we can make. And this is why it might be important to actually divide this. Now, my last recommendations, and then I'm done, and I'm looking forward to your questions and comments. What can I say about this? How can we sum this up? What kind of recommendations can one make? I think it's really important, first of all, especially if you work in the basic research field, that you obtain information about your samples. As I said initially, A lot of times when I ask the question, people don't know the sex of the cells, the karyotypes are not known, even the procedures or the potential effect of the procedures that are being applied is not exactly clear and has not been investigated. So this is definitely something to look out for. When working with populations, especially when working with humans, if we look at the gender aspects here, I think at least both sexes should be included. I showed you the review by Frank Morvey-Jarvis that, to find out if there are differences, you need first to compare the two mice, the female and the male mice. And of course, if you work with humans and you want to look at gender, you should have a clear idea about which dimensions you want to investigate, which dimensions can impact the potential outcomes you're looking for. I think it's always helpful to consult experts. There's quite a few by now in different fields, in different disciplines. They can help you with the state of the art and help you set up your experiments. Of course, it's important to disaggregate data if it wasn't clear enough with the digoxin study showing that stratified analysis is helpful in pointing out potential differences in efficacy and most importantly in potential side effects. What a lot of times what we're seeing is that we might have analysis, but we don't see that in the report in the papers. So this is really an encouragement. Please, if you're looking at these differences, report them. We really need to know. And if it's not reported in the main paper, there's always the opportunity to have supplementary materials. And of course, it's still important to spread the word about this. Several colleagues still don't know about it, are potentially open to the subject, but don't know how to do it. So there are many materials that can also be used. And so it's still a matter of increasing the awareness. And with that, I thank you very much for your attention, and I'm looking forward to the discussion. Thank you.
So Sabine, we have a great question from Sarah Jackson. And you probably can see it if you come off your screen, but I'll read it. Regarding specifying sex and gender in clinicaltrials.gov for COVID trials, how is it that the NIH sex as a biological variable policy is being ignored? Further, it seems that this policy is only followed at the beginning, funding, and perhaps at the end with publication given the Sager guidelines. Do you have any suggestions for how to increase uptake by investigators at different stages of the scientific process?
I think this is an extremely important question. It's something we have been discussing with the EU Commission as well, because it's something that we're seeing in general. So let's take one step back for the NIH, for the database, the FDA database. There is one standard question that you have to tick where it says sex, female, male, all. And what we were looking for, and we specify that in the paper as well, is if it was mentioned apart from ticking the box, which you have to tick, is it mentioned at all in the reports that are being submitted? And there's where you already see the differences. But then I think the question of monitoring is absolutely an essential one. So we're seeing this at the European Commission as well, where again, it is mandatory to report in how far sex and gender will be considered, but then there is not really a follow-up. So we generally have to submit interim reports, so we have to submit some kind of document, and those are really not checked to make sure whether what has been promised, I mean, let's just say people will promise pretty much anything in order to get the money to do the research, whether that has actually been implemented. And again, I would argue that even at publication level, at the end of the granting, at the end of the granting time, at the end of the availability of the money, nobody's really checking that much anymore if it is being reported or not. I think Canada is doing a bit better. Most European countries don't even have these obligations. Some do, and I would say in most cases it's a nice nudge. The Netherlands, Germany, are asking about it a bit more insistently, so you have to say something. But I would again say the problem is it is up to the reviewers to point out if this was sufficient or not. And then again, and so we do incur a lot of times in situations where the grant is wonderful. They haven't done it, but the grant is great. They should still get the money. So this happens. And then again, the monitoring process is not implemented properly. And I think that is really, we all have to submit interim reports unless a grant is for 4 or 6 months, which is really rare. We all have to submit interim reports. And I think that would really be an instrument for the granting agencies to check whether these promises have been kept.
You know, and I'll also add, I'm on the advisory board for the Office of Research on Women's Health, and Janine Clayton is really one of the drivers of sex as a biological variable. They are tracking that. It is improving in terms of people actually getting funding that meet those criteria. But even there, a lot of people are still getting the funding, as Sabina said. There's also an anonymous attendee said, beautiful presentation, which I absolutely agree. That was fantastic. Thank you so much. Could you add something about the role of gender bias in issues like medical gaslighting and how, if there are ways to both measure the impact of this specific issue and the mitigation measures?
Yeah, well, that's an entire, I could give an entire presentation about that. I would say it goes a bit beyond what we're doing today, but of course it's an essential aspect. And as you mentioned briefly in the introduction, I do work in that field as well. And we've worked quite a bit on sexual harassment in the academic field as one of the forms of discrimination. And we're doing several initiatives to try to support organizations in, well, developing preventative measures and supportive measures. It's, of course, an essential topic to investigate. I think it's a really difficult topic to investigate because we are dealing with, I would say there is an analogy to measuring gender when you want to measure gender discrimination in organizations, because again, it is a multi-layered phenomenon which is not easy to quantify. And I think that's a bit of one of the points we're struggling with, apart from the fact that in a lot of organizations, there's not a lot of openness to investigate it, to actually provide numbers, to look into it. But even if there is this openness, assuming that an organization is really proactive and wants to do something, it is not easy to measure. What we usually resort to when we measure it is in quantitative terms, which is complicated in measuring the underlying effects. So I know that here in several of my colleagues here in the Netherlands have done qualitative work. I would recommend the work by Sarah Ahmed, who's done a lot of qualitative work on complaint and discrimination procedures. I think we are still at the point where we also were, I would say, with sex and gender sensitive medicine, where we're still raising awareness. And it's a phenomenon where we know it impacts science, we know it is present, none of us is questioning that it exists, and yet the research into it and the reaction to it, the implementation of preventative measures is really lagging behind. So I think again, there are several analogies, but I think the difficulty in tackling it has to do with resistance, but also with a certain degree of methodological difficulties. And then, of course, the fact that, well, there are structural aspects that don't help in trying to develop preventative measures and in reacting. And I would say as much because I could talk about this for an hour, but maybe we can have a different opportunity to engage in that with a bit more data.
Well, we definitely will get you back on that. I do want to encourage anyone who has questions to put it in the Q&A. We'll get to it. In the meantime, I would like to ask a question. You started in on it. You were showing the BEM scale and you were talking about different, issues about measuring gender. But I think you're aware that there was a Stanford project that on, gender as a sociocultural variable. So I think we've coined that term. Can you say anything about that and what, the challenges, methodological challenges, and then, where we can go with that?
I was hoping that Londa Schiebinger would be here to maybe say 2 words about that. I mean, I know it happened under her tenure, but as I said, she might be.
Yeah, I know that Londa's on the webinar, but we didn't have her as a panelist, which is unfortunate. So we'll do that.
Which would be great to hear about that. I think what they did was what was probably one of the real advancements that we've seen in the last years in the whole discussion about the operationalization is trying to circumvent talking about masculinity and femininity and really bringing it down to the real factors we're actually measuring. I think it is still a challenge because the whole discussion that this opens up is, I think it is methodologically more sound, but it becomes more complicated because at that point, if we break it down to the different the different variables we're measuring, so the different attitudes, for example, is that then still gender? So I think that's the question where at a certain point it becomes more tricky. I think it is correct to measure the single dimensions rather than calling this a bulk of masculinity and femininity and making it into a dichotomous concept. What becomes, I think, a question then in this step is are we still measuring gender, or are we measuring certain traits that are associated with that? So it kind of it opens up, I think, a discussion on whether how should we call this. So I think that's where I see it as a definite methodological advancement. I think it is really important and is much more precise. But I do see a methodological conundrum in bringing that a bit back in, are we still measuring gender, or are we measuring traits, for example? And is that then still gender, or is it not? I mean, in a way, we can also say we are all embarking in a process here. So in a way, we could also say that maybe we will not measure biological sex anymore at a certain point if we know that it is a certain hormonal concentration, or let's say it's a certain genetic asset. Why shouldn't we just measure that instead of calling it sex as a proxy? I mean, that could be a philosophical discussion about, maybe we don't need the terminology at all anymore at a certain point, but now we still need to make clear what it is that we're looking into, what kind of impact that has. And the easiest way to do that, the most direct way to do that, is by working with sex and gender. But maybe in the long run, we can pinpoint the elements that are actually making up the difference, and we will not be talking about this anymore. So I think that's also a question that would be open for discussion.
Well, we have one more question, but before we do that, I'm going to just quickly follow up and say, as you introduce that issue, intersectionality is now something that we're all talking about as well. And so when you start to say gender versus trait, like gender, all the concepts you're saying for gender differ by every culture. Absolutely. And by every race and ethnicity, and by every age group, and by every religious group, and all of those other things. So how will you tackle that issue?
I think that's the next step. In a way, I work together with a few colleagues that work in the field of intersectionality, and I think it is the direction we're all moving into. My choice at this moment in time, it's not to say that it is not important. I think it's the reality, quite frankly, that we are observing in our social environment. But I think we are still not clear enough on a lot of the methodological aspects that we can simply move over to an intersectional approach, I think, for some of these questions that we're asking. So I think there is still work to be done in order to make the concepts that we want to investigate in an intersectional fashion, make those really methodologically robust so that we can take the next step, and then also show their entanglement and disentanglement. And again, I think if you're working with methodology from the social sciences, and we do a lot of qualitative work, you can show the intersection, the entanglement nicely. It's possible with those methodologies. However, in the biomedical field, to increase the acceptance of the topic of sex and gender, you need to be able to work with quantitative methods as well, because this is where most of the research is being done. And I think we need to be sure that we have sound methods here so that then we can implement intersectional analysis also in quantitative fashion, where we're seeing some being done, but I think there's a lot of room for future research and exploration.
Which leads perfectly to the last question, and we only have 2 minutes. Carolyn Thorne asks, how can databases help push people towards best practices for collecting and reporting?
Yeah, I do think that for the databases, well, it depends a little bit which kind of databases we're talking about. So I think in single studies, you have the opportunity to go a bit beyond just asking about binary sex. So I think it really depends a bit on the researchers involved. Although we have to say clearly, and these are discussions that I had 2 days ago in a conference I was in Europe, is about if you go ahead and let's say you do it in a more innovative form, in a more complex form, how do you still compare your data, for example, and this was a large European consortium speaking, how do you compare then your data with the data of the others? So in a way, you're trying to push forward, but you have to make sure that all the others agree as well, especially if you want to compare data. So this is a bit of a conundrum where on the one hand side, you can go forward if it's your database, in quotation marks, but of course, if you want to compare, it's difficult. If it's statutory data that is acquired at the national level, that is acquired at the federal level, the agencies do have the opportunity to take steps forward. And for example, we're running a project in Germany at the moment where we're working with the statutory agencies and trying to figure out how far can we go and move beyond a binary sex question, and make sure that it is, on the one hand side, you're not allowed to ask too many questions because questions are time, especially for statutory surveys. On the other hand, you want to acquire information about diversity. And on the other hand, you also need to make sure that you still keep a priority on the bulk of the population that you're trying to address. So you have to navigate potentially conflicting questions that are coming together in, I want to make this more, well, let's say I want to do this in a fashion that's appropriate for 2022. And on the other hand, I need to compare with other European data. And on the third, I need to ask questions that people understand or can identify with. So there are many layers that we're addressing there. And just to say, things are moving a bit forward, but there are also all these questions about comparability of databases, about moving forward with others that slow the process down a little bit.
Well, fortunately, Alaya answered a question from another person, so from Jonathan. So thank you, Alaya. And I think, unfortunately, we've run out of time, but it's clear that we should do another one of these and get Londa Schiebinger on here with you. Yeah. And we will set that up. But everybody, we will be posting this on the Wisdom YouTube channel. So we have recorded all of our talks over the last 8 years. And so go to the Wisdom website to get access to that. We probably won't have it posted till early next week because there's a little bit of work that needs to be done. But that was fantastic. And thank you so much, Dr. Oertelt-Prigione. I hope I said that right, but I'll call you Sabine if that's okay.
Yeah, please go ahead.
Okay, wonderful. Thank you so much.
Thank you.